Thank you, Heather. So as Heather stated, my name is Karin Knudson, and I am a Chemistry, Manufacturing, and Controls Reviewer, or CMC Reviewer, within the Office of Therapeutic Products in CBER. Today, I will be talking about CMC considerations for early phase studies, specifically for cell and gene therapy products. So by the end of this presentation, you should understand why manufacturing investigational cell and gene therapy products presents unique challenges. You should be able to identify the CMC information you need to include in your early phase IND submission. And you should be able to describe some common CMC issues that arise during product development and also identify some approaches for avoiding them. So within OTP, we regulate a diverse product portfolio, which includes gene therapy products such as genetically modified cells like CAR T cells, viral vectors, and tumor vaccines. We also regulate cell therapy products like stem cells, anti-tumor and antiviral T cells, and xenotransplantation products. The diversity of products we regulate means that we encounter different manufacturing paradigms than typically seen with conventional drugs or biologics. With conventional drugs or biologics like vaccines or monoclonal antibodies, typically one product lot is manufactured to treat many patients. But in the case of cell and gene therapy products, we usually encounter the situation that one product lot treats a few patients, or in some cases, one product lot will be manufactured to treat only a single patient. This difference means that sponsors will encounter many unique issues during the development of these products, including the use of advanced manufacturing techniques, issues with scale-up of manufacturing and distribution, and typically the impact of a manufacturing failure is greater for cell and gene therapy products. Because of the diversity of products we regulate and the different manufacturing paradigms and issues we encounter, the regulatory review of cell and gene therapy products is highly product dependent. For example, for a specific type of product, one lot could treat thousands or just one patient. Sponsors use a wide variety of manufacturing procedures and techniques to manufacture their products. We also see that products that have little test material or a short shelf life will present challenges with comprehensive testing, and that is generally handled on a case-by-case basis. I do want to make the point that the risk of each product varies because that risk is dependent on the source material and how the product is made. Finally, some of our products are highly variable, especially in the case of an autologous product, which makes demonstrating manufacturing consistency and comparability challenging. That again will need to be handled on a case-by-case basis. So given that the regulatory review of cell and gene therapy products is highly product dependent, how do you actually develop your products, and what is paramount to tackle early to avoid review issues? So overall, the stage of product development guides the review concerns, but safety is the primary concern at all stages of the product development lifecycle, regardless of whether you're coming in for a phase 1 study or submitting a biologics license application. As you progress through the product development lifecycle, you will need to start focusing on different aspects of product development, such as product characterization, qualification and validation studies, and assessing product potency. But overall, product development does tend to occur on a continuum, and as you learn more about your product during clinical studies, our expectations will increase. I do want to emphasize that product safety and quality do not happen by chance. Safety and quality need to be designed into the product, so you should start this process early. It is especially important that you start product development early because cell and gene therapy product development should progress in parallel with clinical development. We have seen sponsors struggle to keep up the pace of their CMC development compared to their clinical development, which we all know can happen very rapidly nowadays. Just to reiterate, CMC expectations do increase through the product lifecycle, so you need to stay on top of this. But today, we're focusing on early stage INDs, so let's discuss what chemistry, manufacturing, and controls actually are. So chemistry, manufacturing, and controls, or CMC information, covers everything about how you manufacture your product, and it should describe how you ensure the safety of the source materials, intermediates, and final product. So this information includes details on product manufacturing, details about the raw materials you use, the manufacturing process and the quality systems, and product safety and quality testing, either in process or release testing. This information also includes product stability, storage and shelf life, what container you use, what label you use, how you track your product. In some cases, you may be able to cross-reference related INDs or master files to provide some of this information, but documentation of that cross-reference is needed. Overall, the CMC information provided in your IND should demonstrate your capability to consistently and reproducibly manufacture the investigational product. It should include information that describes the composition, manufacture, and control of the investigational product, and it should be sufficient to assure identity, quality, purity, and potency or biological activity of the investigational product. So diving more into this. As stated earlier, early phases focus more on safety and specifically knowing the identity, purity, and activity of your product. The amount of information that you will need to submit under your IND will depend on the phase and the scope of the initial clinical investigation. And as development proceeds, it will be necessary to supplement the initial CMC information to address the expanded phase and scope of your clinical investigations. And as you move on to later phases, we will require more information on the quality and potency of your product. So what are some of the common issues that we see with early phase studies, and how can you avoid these CMC issues with your submission? Remember, we are primarily focused on product safety here. If you are using donor-derived materials, you should describe how donors are tested to ensure the source material is safe. Sometimes sponsors don't include sufficient information about the release assays that they perform to ensure safety of their product. So make sure you include comprehensive information about your release assays. In particular for safety assays, such as those assays for sterility and mycoplasma, you should describe the methodology, and in general, safety assays should be qualified for their intended use for early phase studies. Determining the dose of your product is really important, so you may need to qualify that assay for its intended use. We also see that some sponsors don't include sufficient information about the reagents they use during manufacturing. You should include the source and documentation for each reagent, which is typically provided as a certificate of analysis, and that information will be used to show that each reagent is suitable and safe for use. One final consideration I will mention is that you should document the delivery device you intend to use to administer your product, including its regulatory status, and provide data demonstrating that your delivery device is compatible with your drug product. While I talked about quite a few safety issues you should consider and address in your IND, please be aware that this is not an all-inclusive list. I highly recommend that you read the FDA guidances about the CMC information you need to include in your IND. There is a gene therapies guidance and a cell therapy guidance, and both contain very helpful information. But what should you do if you have more questions? If you have more questions for us, I highly recommend you take advantage of the meeting opportunities that Heather went over in her presentation. In particular for early phase studies, the Interact meeting and the pre-IND meeting should be used to discuss any unique issues you are running into or any questions you may have regarding your product prior to submitting your IND. There are other meeting opportunities for later phase studies, but since this is an early phase study discussion, please consider an Interact or pre-IND meeting. So finally, to summarize what we talked about today, the CMC information submitted under early phase INDs should demonstrate your capability to consistently and reproducibly manufacture a safe investigational product. The regulatory review of cell and gene therapies is highly product dependent, so we do recommend that you talk to the regulators, which is FDA, about challenging issues or novel approaches you plan to use. Safety and quality must be designed in the product, so I'm going to stress again, start this process early. This slide provides contact information and a lot of links to websites that contain valuable information on CBER INDs and how you can interact with us. That concludes my presentation. I will now pass it on to Diva Veena.